companies to assess whether the product is worth a larger scale investment of time and resources. For those drugs that show sufficient promise, Phase 3 studies are designed to provide evidence of the product's efficacy by enrolling hundreds or thousands of patients as research participants. With results from all 3 clinical trial phases, pharmaceutical companies can then seek approval to market their drugs. If approved, the FDA or other countries' regulatory bodies might also require post-marketing surveillance studies called Phase 4 trials in order to have more safety or efficacy data about the product, even as it is widely prescribed to patients. Given the complexity of the drug development process, a clinical trials industry consisting of companies that assist in the design, management, and analysis of these trials now exists and profits from pharmaceutical companies' efforts to get new drugs to market as quickly as possible. This book focuses exclusively on Phase 1 clinical trials conducted on healthy volunteers. In most therapeutic areas, healthy volunteers are the preferred research participants for these safety studies. However, terminally ill or disease-affected patients are also sometimes used. In lay terms, Phase 1 trials measure the body's changes or adverse events, AEs, that occur when research participants are given an investigational drug. Starting with small doses and increasing the number of milligrams over the course of a trial, investigators assess the tolerability of the drug as they seek to find a therapeutic dose without burdensome side effects. After administering an investigational drug to participants, researchers document all physiological changes that are captured by medical procedures, as well as participants' self-reported symptoms. These changes can and do include a range of experiences that researchers suspect are not caused by the investigational drug, which is why the preferred term is adverse event rather than adverse effect. AEs, such as headaches or stomach upset, are common occurrences, and they prove ambiguous with regard to causation. Researchers must then adjudicate, along with the pharmaceutical company sponsoring the trial, which adverse events are or could be actual adverse drug effects. Given the goals of Phase 1 trials, the advantage of enrolling healthy participants over patients is threefold. First, there is less ambiguity in the interpretation of AEs when there are no underlying illnesses that create symptoms in participants. Second, the risk of serious complications is diminished when participants have normal kidney and liver function and can withstand temporary impairment to these organs. Finally, and arguably most important, the recruitment of healthy volunteers tends to be very rapid, especially compared to studies requiring ill or diseased participants. And efficient recruitment helps to speed successful products through the development pipeline and onto the market. Phase 1 trials are distinct from later phase studies, that is, Phase 2, 3, and 4 trials, not only in the enrollment of healthy volunteers, but also in the study of the study protocols. Phase 1 studies tend to be conducted in residential clinics that specialize in this type of research. These studies are also shorter in length, lasting only several days or weeks compared to several months or years for later phase trials. And they generally require participants to consent to a clinic confinement for some part of it, if not the entire study. The confinement period enables control of participants' diet and frequent collection of data about the investigational drug.
To keep listening to this audiobook, I've left you a link in the description so you can do it, at no cost thanks to Audible's 30-day free trial.